It's a pleasure to welcome you all here tonight to the first lecture in the 2019 UOW Knowledge Series. I'm Dr. Luke McCallery, and I'll be your MC tonight. I'd like to introduce to you the main event, the guest speaker, Professor Justin Yerbury. So, a little bit of background on Justin. He first heard about MND around 20 years ago when his uncle, cousin, and mother were diagnosed in quick succession. It was clear at the time that there was not enough understanding of the molecular basis of the disease to figure out what the cause was, or even, most importantly, find a cure. As a result, Justin decided to dedicate his life to increasing the understanding of both the origins of motor neurone disease and also trying to figure out ways to potentially slow down or cure the disease. To do this, he went to the University of Wollongong to study, and he completed his PhD in 2008, after which he was awarded an ARC fellowship to undergo a postdoctoral research fellowship with Professor Christopher Dobson at the University of Cambridge in the UK. Later, in 2009, he was awarded the eminent Bill Gohl MND fellowship. And then later again in 2011 was awarded the Vice Chancellor's Emerging Researcher Prize. In 2012, Justin was awarded an ARC DECRA fellowship to build his own research group around protein homeostasis defects in ALS MND, which we'll hear about tonight. And later, in 2015, he was awarded an NHMRC Career Development Fellowship to continue building this team within this area. 10 years after graduating with his PhD, Justin was appointed as UOW's Professor of Neurodegenerative Disease to reflect his contributions to the fields of protein homeostasis and MND. He was also recently awarded the Betty Laidlaw Prize from the Motor Neurone Disease Research Institute of Australia for his contributions to the field in 2018. And then just this year, in 2019, he was awarded Citizen of the Year at Wollongong's Australia Day Awards. And now I'd like everyone to give Justin a round of applause as he comes up to the stage.
Good evening. Thank you for the kind introduction. Firstly, I would like to thank the organizers of the lecture series for inviting me to talk to you here tonight. It is always an honor and a pleasure to share my thoughts and the work that we do. Secondly, I would like to thank my dedicated team of staff and students who are the ones that do all the work in the lab. The story I would like to share with you tonight will give you, I hope, an insight into the reason why we are pursuing a particular line of research and a new appreciation for proteins more generally. I have titled this lecture Proteins, a story about the stuff of life and how it shapes our understanding of motor neurone disease. So I will first address my claim that proteins are the stuff of life. I will give you a few thoughts and ideas about proteins and tell you why I think they are important. First, we have to understand the answer to the question, what is life? Life is something that philosophers have argued about for millennia. Our modern biological understanding of life remains complicated, but the general consensus is that there are several characteristics that define living things. There is not just one distinguishing feature that separates a living thing from a non-living thing. A cat moves, but so does a car. A tree grows bigger, but so does a cloud. A cell has structure, but so does a crystal. Biologists define life by listing characteristics that living things share. Something that has all of the characteristics of life is considered to be alive. The 7 characteristics of life include responsiveness to the environment, growth and change, ability to reproduce, have a metabolism, maintain homeostasis, being made of cells, and passing traits on to offspring. This can be best illustrated by comparing cells from any living organism to a virus. Although controversial, many do not consider that viruses fit the definition of life. In particular, viruses do not respond to stimuli, they do not metabolize, and they cannot reproduce on their own. Viruses are simple particles that consist of inheritable material, either DNA or RNA inside a small shell. Some viral DNA contains as few as 14 genes. In comparison, human DNA is thought to contain about 30,000 genes. Although some would consider DNA to be the molecule of life, it is a static molecule, and this explains why a virus cannot perform those activities we associate with living things. While DNA provides the information required for the characteristics of life, the fact that the actual DNA molecule itself is quite static means that most of the actual processes that define life are carried out by proteins. Even inheritance of genes cannot take place without proteins. You can think about proteins and DNA like the chicken and egg scenario. You cannot have one without the other. If a cell divides and produces a daughter cell that contains the DNA with instructions to make all the proteins necessary for life, but no protein molecules to facilitate the making of new proteins, the cell will not survive. Inheriting a set of proteins that can make new proteins is vital then for life. Now, back to our viruses. To reproduce, a virus must hijack the cell's machinery that makes proteins and that can reproduce its DNA. Much of this machinery are made up of proteins in the host cell. So if we believe that viruses do not fit the definition of life, the thin line between life and the non-living is not whether or not an organism has DNA but whether it contains the molecular machines of life that allow it to metabolize proteins. Let's now turn our attention to looking at what exactly is a protein. You might best know protein as a vital part of your diet. You might even know that some foods are rich in protein, like red meat and egg white. But proteins are much more than something we eat. Proteins are an important part of every cell in your body and indeed every living cell on the planet. Even plant cells, which are not renowned for dietary protein, rely heavily on their proteins for life. To be more specific, a protein is a molecular machine that is made from the instructions stored in your DNA. So, proteins are the things that bring to life the genes we inherit from our parents. The proteins that are coded by your genes are essentially molecular machines that do vital work in all parts of your body, and include things like hemoglobin to transport oxygen, enzymes to digest food, collagen to provide physical structure. In addition, other things like hormones, immune systems, and electric signals in the brain are all also controlled by proteins. Humans have the instructions for about 30,000 different proteins stored in their DNA, providing an amazing array of molecular machines available to the body to maintain the complexity of life. Proteins are able to do all these jobs because each protein has its own unique three-dimensional shape that has evolved to allow for a very specific function. You can see here colored reconstructions of a range of proteins. They may just look like blobs, but the subtle differences that we can see are exquisitely defined at the atomic level and allow each protein to perform its own very specific job. There are different ways of displaying proteins, and here I am showing you what is called a space film model. Throughout this talk I am going to try and show you representations of proteins like this that are as accurate as possible. These protein shapes are not visible to the naked eye. In fact, they are not visible on our most powerful optical microscopes. In this image of neurons taken on a confocal scanning microscope, each pixel is 50 times bigger than a protein molecule. In order to see protein molecules at the highest resolution, we need to use something more powerful like an electron microscope. And here in Wollongong we have one of the most powerful available. So there are exciting times ahead at the University of Wollongong discovering how proteins work at the most fundamental level. To tell you that something is 5 nanometers in size may not mean much to you. To give you an idea of the size scale on which proteins exist, let's work our way up to compare to something on a scale that you are familiar with. An average protein is about 5 nanometers in diameter, and a small virus particle is about 6 times bigger than that. A single staph bacterium is about 20 times larger than that again. A red blood cell is 10 times bigger than that. And another 10 times larger than that is the width of a large neuron. A frog egg is around 10 times larger again at 1 millimeter in diameter, and I think we are all comfortable with millimeters. That means that we could place a protein on and about 200,000 times across the diameter of a frog egg. While the proteins I have shown you look like solid objects, it is important to remember that protein exists in the watery world of the cell. We say that they are in solution or soluble. They are dissolved in water just like salt dissolves in water. The best way to imagine what proteins in solution looks like is to think about egg whites, which is basically a concentrated protein solution. When there are so many proteins in solution, it becomes quite viscous. I have told you that proteins are molecular machines, so here is a working example of one particular protein. This is ATP synthase, and it acts like a generator converting an electric charge across the membrane into biological energy used by other proteins in the cell. This is so important that without this protein there would be no life. the function of the ATP synthase, and all proteins for that matter, rely on their precise three-dimensional shape. We call this shape a fold, because a protein is made as a linear chain of amino acids and has to fold up into its shape to do its job. You can think of this process just like a coat hanger. It is made from a linear piece of wire, and it is then folded and twisted into shape before you can use it for its function, to hang a shirt. A protein's fold is so important that it shapes evolution. To study evolution, scientists usually create evolutionary family trees, or phylogenetic trees, by comparing genes between different species. The more genes two organisms have in common, the more closely they are related. But this technique only lets you rewind a million years or so. Any further, and by the end A has mutated so much it is impossible to see the similarities between species. Some scientists claim to have examined evolution back to the beginnings of life on Earth, billions of years ago. Not by comparing genes, but by comparing the shape, or folds, of proteins. If you change the shape of a protein, you disrupt its function. Because not every genetic change will alter the fold of a protein, life can tolerate a continual gentle drift in the genetic code, but protein shape is critical and therefore evolves much more slowly. So again we see the surprisingly important role protein shape is playing, this time behind evolution. Hopefully, you are beginning to see why I have said in my title that proteins are the stuff of life. Proteins are so important that it is the combinations in which they are made that defines various cell types in multicellular organisms like humans. While the DNA of an organism stays relatively stable across the billions of cells in your body, the level of each of the 30,000 proteins can vary. This means that the potential number of cell types is limitless. You can think about this like a huge audio mixer, where you can dial up or down 30,000 different channels, and each combination will give a particular sound, but in this case it is proteins and each unique combination gives a particular cell type. For example, red blood cells have high levels of hemoglobin, where all other cells have none. Another example is the MND-associated protein called SOD1. It is found in every cell in the body but is most abundant in liver and in some parts of the brain. This type of comparison can be made over and over for up to 30,000 different proteins. To help understand the fine line between living cells and viruses, some scientists in Switzerland determined that there was about 500 or so proteins that a virus will hijack in cells in order to reproduce. They found that proteins that either help make new proteins, remove or degrade other proteins, and those that transport other proteins to particular parts of the cell are the most hijacked. This makes sense because viruses have no way of making or recycling their own protein. Using the same cellular processes that viruses hijack, each individual cell has the job of making sure that the level of each protein is just right for the job it has to do in that cell. We call this phenomenon protein homeostasis, where homeostasis means the tendency towards a relatively stable equilibrium, especially as maintained by physiological processes. So the particular combination of proteins in each cell is exquisitely balanced. So when we talk about protein homeostasis, we are talking about keeping all proteins made by a cell in a steady state. Which means making just enough protein, in the right shape, at the right place, at the right time. This task is mind-boggling when you consider that there are more individual protein molecules in an average human body than there are stars in the universe. Here you can see me explaining this idea to eminent theoretical physicist Stephen Hawking. He obviously agreed with me on the importance of proteins as he agreed to officially open the Center for Misfolding Diseases in Cambridge, which aims to find cures for diseases that are associated with protein deposits such as Alzheimer's disease, Parkinson's disease, and MND. If you still cannot grasp the vastness of the number of protein molecules in a human body, think of it like this: there are more protein molecules in the human body than stars in the universe, and it has been suggested that there are more stars in the universe than there are grains of sand on every beach on Earth. And remember, all of those proteins in your body need to be monitored and kept in check. It is this protein homeostasis framework that is allowing us to further understand motor neurone disease. Keeping the fine balance of protein levels in a motor neuron is like walking a tightrope, and just like a tightrope walker, the only thing that saves us is balance. Motor Neurone Disease, or MND as it is often called, is a term that describes a range of debilitating and progressive neurodegenerative diseases. It is often mistaken for a musculoskeletal condition because of the obvious wasting of voluntary muscles, but in fact is a result of the death of the neurons that control muscle movement. Without their connection to the brain, the muscles waste away. Amyotrophic lateral sclerosis is the most common and rapidly progressive form of MND, and it is not as rare as you might think, with a 1 in 400 lifetime risk of developing this condition. This is the form of MND we study here. In most, but not all, cases of MND, cognitive capacity is not affected. Following the onset of MND, approximately 50% of cases result in death within 30 months, while 20% can survive from 5 through 10 years, with rare examples of people living more than 50 years after diagnosis. Stephen Hawking shared with me his thoughts about his extreme longevity, but in fact since the 1980s, Stephen was at least partially mechanically ventilated, and it is respiratory failure that is the most likely cause of death in people with MND. The brain is a complex network of neurons and their supporting cells. In total there are tens of billions of neurons in the brain, but not all neurons are made the same. There are particular neurons that lay down memories that are different to those in charge of sight or hearing. These distinct neurons not only look different but respond differently to chemical signals in the brain. There are many types of neurons for specific tasks in the brain. The differences can be explained by the distinct pattern of protein levels unique to each neuron type. This is important because specific neuron loss is responsible for the differences between neurodegenerative conditions, Alzheimer's disease, Parkinson's disease, and MND. It is the understanding of why very specific neurons die while others remain resistant to disease that has puzzled researchers for decades. So understanding why motor neurons die in MND is a question that is on the top of the list of things we need to know. Because of this, many people have asked what are the characteristics of motor neurons that makes them distinct from other neurons. The first thing to note is that motor neurons are themselves relatively rare, a few 100,000 in a sea of billions. The next thing to note is that they are very large. Not only do they have a large body, but they are among the longest cells in the nervous system. They can be upwards of a meter long. This large size means that making sure that protein balance or homeostasis is a particularly unique challenge. There is a cost to transporting proteins over a meter to perform their function. This is an energetically expensive exercise. Much like the cost of fuel is always more expensive at remote locations. The job of keeping all these protein molecules in balance is a gigantic task, and sometimes it can be too much for a cell to deal with. In this case, proteins will accumulate into what we call aggregates. These are deposits of proteins that are normally in solution, or soluble, that lose their shape and unravel or unfold and crash out of solution. So while most proteins are soluble, the deposits form a more solid-like state. I am confident almost everyone in this room will have done an experiment to see what it takes to make proteins in solution aggregate. Here is the experimental outline. Step 1. Take your protein solution. And step 2. Alter some amount of stress until the proteins unfold and aggregate. I am hoping that some of you recognize this experiment to be frying an egg. As I mentioned earlier, egg white is a concentrated protein solution, and if we impose just enough stress via heat, the proteins will unfold and aggregate. This changes the proteins from a transparent solution to an opaque solid. Now I am going to show you what this process looks like in a single cell in culture. Here on the right is a movie that follows an aggregate forming in a cell, which you can see as a bright spot becoming bigger and bigger, and eventually what you see is that this cell dies in what looks like a little explosion. In this next video there are hundreds of cells in each image. To the left are cells with a genetic form of MND, and to the right are cells that look genetically modified to fix the protein. You can see the bright spots forming in the MND cells. We think that these protein aggregates are detrimental to cells. Just as spontaneous aggregation of egg white proteins would mean loss of nutrients to a growing chick, it is likely motor neurons lose important proteins in the aggregate. Deposits don't occur in healthy people without MND, and it is likely that motor neurons stay healthy until just before disease onset. So it makes sense there must be a trigger that causes stress to motor neurons. Our way of thinking is that anything that upsets the fine protein balance could potentially lead to protein accumulation and deposition. With tens of thousands of proteins spread over a meter, it is amazing to think that things can't go wrong more often. It is thought that evolution drives a protein to be more soluble when it needs to be at high concentration in the cell. So proteins that are only required at lower concentrations are less soluble. Evolution will pressure proteins to be soluble enough for their jobs but no more soluble. So there is not much give in the system. Our proteins are living life on the edge under normal circumstances, and it does not take much to tip them over the edge. We have been trying to understand why motor neurons are particularly at risk of having protein deposits form. And we think it has to do with the unique combination of proteins and motor neurons. Our analysis has identified that the cells that die in MND, called motor neurons, are particularly at risk because their proteins are supersaturated. Supersaturated means that there are so many more protein molecules in solution than what we might expect. You can see from the picture on the left that the inside of neurons are very crowded. And this is only representing 65 proteins out of possible thousands. Supersaturation is a word used more generally to mean that a solution has more of a substance in it than possible under normal circumstances. This means that if conditions change, the substance is at risk of crashing out of solution. The most common examples of supersaturated solutions are salts in water. Usually a little heat will help more salt go into solution, but it is then at risk of precipitating, like the supersaturated solution of sulfur here that crashes out of solution and forms crystals. We think that this concept applies to the proteins of motor neurons where the proteins are ready to fall out of solution or aggregate when conditions change. So what kinds of things can trigger protein imbalance in motor neurons? We know that approximately 5 to 10% of all cases of MND are inherited and caused by faulty genes. Natalie has shown in experiments that even one small mutation in specific genes is enough to cause protein deposits form. These diagrams represent 3 different genetic forms of MND and each have deposits. In this case, the arrows point to the deposits. The remaining 90% of cases are random or sporadic in the population, and there is no consensus on what the underlying cause of these cases are. 2 intriguing possibilities are a toxin from blue-green algae and reactivation of ancient endogenous retrovirus in our genes. The idea of how these may trigger protein imbalance is just as intriguing. The toxin works its way into proteins, which makes them change shape and malfunction, while activation of dormant viruses our ancestors acquired millions of years ago would put pressure on the cell systems that control protein production and degradation, allowing protein deposits to form. While some cases may be associated with these factors, there is still much work to do to understand the disease trigger in sporadic MND. An important part of what we have found is that when aggregates do form, one thing that motor neurons do to respond is that they slow down the production of the proteins that are at most risk, the ones that are supersaturated. By making less of the at-risk proteins, neurons can make sure that there are less proteins aggregating. However, this strategy used by the cell is like turning off your water at the mains. If the bathtub is overflowing, it will stop the barred water, but will also mean lots of other taps in the house won't work. In motor neurons, some of the most at-risk proteins are in charge of controlling electrical signals. They do this by moving charged molecules across the boundary of the neuron. On the left you can see an example of one of those proteins building up a calcium gradient. By slowing down the production of all proteins at risk, this means that many of the proteins that control electrical signals are reduced. If you look at the graphs on the right, you can see the red bars represent less than normal production and green is an increase in production. This tells us that most of the proteins important for controlling electrical signals are reduced. This reduction leads to the motor neurons not firing properly, which can be measured in people with MND, and which in some people with MND is thought to result in muscle fasciculation or twitching, as seen here in this person's leg muscle. These motor neurons are thought to be the ones that go on to die. So our work suggests that genetic mutations that cause MND tips the fine protein balance, resulting in the deposition of supersaturated proteins and motor neurons. Motor neurons respond by turning down at-risk proteins, including those important for electrical signals. Now that we have gained a little insight into what is happening in motor neurons, we have started to think about ways that we can restore the protein balance. One of the things that we are doing is we are trying ways of removing the proteins that are accumulating. We are trying to supercharge the cell's own protein recycling system to remove at-risk proteins. This project has been made possible by the generosity of the community that have raised $30,000. Thank you again all so much. We will begin this project this year. We are also searching for small molecule drugs that may stop proteins from aggregating in the first place. This diagram shows some drugs we have been working on, including copper ATSM, that we hope will stop one of the MND proteins from aggregating by keeping it in the right shape. We hope to test some of these drugs in preclinical mouse models soon. Lastly, I want to thank again all of my staff and students that do all the work in the laboratory, and all of my colleagues here and abroad for their support and collaboration. And thank you all again for listening.